Hi everyone, it's Dr. LeBlanc with Unit 7C, Chemical Changes in Nucleotides. Our learning goals are to understand some of the many different kinds of DNA damage that can occur, to understand the difference between spontaneous and induced mutations, to understand that bases can detach from nucleotides, to understand the consequences of deamination of different methylation states of C residues in DNA, to understand the consequences of tautomeric shifts in bases on base pairing, to understand how substances that disrupt the DNA helix can cause mutations, to understand how base analogues can induce mutations, and to understand the effects of ionizing and non-ionizing radiation on DNA structure. Spontaneous mutations result from abnormalities in cellular or biological processes, normal processes, such as when you have an error in DNA replication. Induced mutations are caused by chemical or physical environmental agents, also known as mutagens. Spontaneous mutations can be caused by depurination, deamination, tautomeric shifts, aberrant recombination, aberrant segregation, errors in DNA replication, insertion of transposable elements, and toxic metabolic process. We're going to talk about these three in this lecture. Depurination occurs when the base is separated from the sugar and a nucleotide that's already incorporated into a strand of DNA. So the sugar phosphate backbone remains intact, but the base has been separated from that sugar right there. Okay, that's known as an apurinic site. All right. When you get to the point of replicating the DNA, this template strand is okay. You have a C residue here, so you're going to put a G opposite. Okay. But if you use this strand as template down here, this purple strand as template, what are you going to put opposite here? Well, there are DNA polymerases that will just put any nucleotide opposite. Okay, so the correct nucleotide to put opposite this, it's supposed to have been a G, but the G is missing, so the correct nucleotide would be to put a C, but you could also put an A, a T, or a G with equal probability. So 75% of the time, you're going to have a mutation by putting a random nucleotide in this position. Sometimes the amine group on cytosine is removed. It's plucked off. That's called deamination. When that happens and an oxygen replaces that amine group, you end up with a uracil base. Now, uracil is not supposed to be in DNA. Uracil is supposed to be in RNA. So when the surveillance mechanism inside the cell recognizes that a uracil base is in the middle of a DNA nucleotide sequence, the DNA repair enzymes can recognize this as an inappropriate base. They remove it, and therefore this does not usually cause a mutation. Now, within DNA, we know that cytosine residues are often methylated. They are methylated as a mechanism to control gene expression and to heterochromatize certain regions of the DNA. So this is a common occurrence in 5-methylcytosine residue in DNA. What happens if you remove an amine group from a 5-methylcytosine residue? Well, you don't create a uracil residue, but instead you create thymine. Now, cytosine is supposed to be base paired with guanine. Thymine is not supposed to be base paired with guanine. It's supposed to be base paired with adenosine. So within the DNA, now you have a GT base pair. 5-methylcytosines create hotspots for DNA mutation because the DNA repair mechanism cannot tell which nucleotide to remove in a GT base pair. Is it the T that you've just created because you deaminated 5-methylcytosine? Or is it the old G that was base paired originally to the 5-methylcytosine? The repair mechanisms cannot discriminate which was the old and which was the new nucleotide. So there's a 50-50 chance that you'll remove the wrong one and create a mutation. Tautomeric shifts involve a temporary change in base structure. Here's a normal thymine-guanine. Oh, here's a not a normal base pairing. Here's a tautomeric shift that creates a thymine-guanine base pair. Okay, and here's another tautomeric shift that creates a cytosine-adenine base pair. These are not the normal base pairs that you would see in DNA. They're a consequence of shifting the electrons around and having abnormal base pairing. Missed base pairing due to tautomeric shifts often leads to mutations because it creates distortions in the helix. These are some of the tautomeric shifts that can occur for each different amino acid, each different nucleotide. The keto form can shift to the eno form for guanine, adenine, thymine, cytosine, and when that happens, you'll end up with missed base pairing. Also in the cell, you can find some reactive oxygen species that are generated by normal metabolism. These reactive oxygen species are just a normal consequence of being alive, a cell being alive. However, they can create oxidative stress that causes DNA damage. So here's a normal guanine base, and here is 8-oxoguanine. Has an oxygen where it's not supposed to be. That's a damaged base, and it can cause mispairing and misaligning of the base pairs within the double helix, which is then the substrate for DNA repair mechanisms. How can we induce mutations? Sometimes we want to induce mutations in the laboratory if we're studying a certain cellular process. Other times, mutations are induced by the environment. Some mutagens that we are exposed to can lead to human cancers. Mutagens can also cause gene mutations that have harmful effects in future generations. And there are mutagenic agents that are usually classified either as chemical or physical mutagens. We're going to talk about both. Mutagens alter DNA structure in different ways. It depends on the type of mutagen, either a chemical or physical. Chemical mutagens come in 3 main types. They are intercalating agents, such as ethidium bromide, which we use in the lab, and benzopyrenes, which are found in cigarette smoke. They can be base analogues, and they can be base modifiers. What about these intercalating agents? What do? Well, they distort the double helix. Here's a diagram showing a normal B helix of DNA, nicely stacked bases, nice smooth sugar phosphate backbone. Some intercalating agents are ethidium bromide, acridine orange, actinomycin D. What do? They squeeze in between the bases. They intercalate in between the bases. They allow us to detect DNA, in the case of ethidium bromide, when ethidium bromide is intercalated in between the helix of the DNA molecule. It emits a glow when subjected to ultraviolet radiation, so it can be useful. However, this distortion can lead to mutations. It's a little bigger picture for you. Distorted DNA is the target of DNA repair mechanisms. DNA repair mechanisms see this distorted helix and say, there's something wrong, we have to fix it. A correct nucleotide, okay, remember this is not changing the sequence of nucleotides, so a correct nucleotide may appear to be mispaired because of the distortion. It can be removed even though it's actually the correct nucleotide, and because of the distortion and the mispairing of the bases, it can be replaced by an incorrect nucleotide. Ethidium bromide is actually a very, very effective mutagen. It can change the sequence of DNA quite readily. Here's another intercalating agent, benzopyrenes. Benzopyrenes are found in cigarette smoke, and they have the same mutagenic effect on DNA that ethidium bromide does. Okay, what about base analogues? Base analogues incorporate into daughter strands during DNA replications. Here's an example of a base analog called 5-bromouracil. The keto form base pairs with an adenine. The enol form base pairs with guanine. So here we have a base analog that can pair with either adenine or guanine. If you feed it to a cell, it will be incorporated into its DNA as a base, and then subsequent to that, it can cause mutations. Okay, so here's our strand of DNA. We have opposite this adenine, the base analog, which was fed to the cell and incorporated into DNA. So this is our starting point. We have our 5-bromouracil incorporated into a parental strand of DNA. Now we're going to replicate these two. Now, A normally pairs with T, and that's okay, but what happens if you shift those electrons around in the 5-bromouracil base? Remember, they can pair base pair either with A or with G. So now you've replicated this strand of parental DNA, you've replicated and put a G where there used to be an A. Okay? And that subsequent change can be perpetuated on and on and on throughout the generations. So you've created a mutation in the nucleotide sequence. What about physical mutagens? They come in two main types: ionizing radiations, like the x-rays you get at the dentist's office, or if you're old enough to have a mammogram, when you have mammograms. And also non-ionizing radiation, which you can get during spring break. So here's a different one slide that shows all the different types of damage to your DNA that radiation can cause. Some of these are caused by ionizing, some of them are caused by non-ionizing. But you can break the hydrogen bonds between your bases. You can have double-stranded DNA breaks in a strand of DNA. You actually physically break the backbone. That's a very severe consequence of radiation damage. You can lose a base, you can change a base, you can have a pyrimidine dimer, you can cross-link other bases together, and you can have single-strand breaks, and you can link proteins covalently to nucleotides. So these things are all not good for your DNA. Ionizing radiation can have direct effects. You can actually physically break a strand of DNA. You can also have indirect effects by causing water to cause damage to your DNA molecules. Non-ionizing radiation is ultraviolet light, the kind that you get at spring break on tanning booths, or if you're a lifeguard and you have a lot of exposure to ultraviolet light. This causes two neighboring thymine residues to covalently attach to each other. Here we have them separated, and now they're attached. This is called a thymine dimer. And a thymine dimer a single thymine dimer in a length of DNA will prevent DNA from being replicated. So it's a pretty severe consequence for your DNA. Mutations can occur spontaneously through errors in DNA replication or can be induced by environmental conditions. Damage to nucleotides can cause genetic mutations. Depurination is the removal of a purine base an A or a G from a nucleotide, with a DNA molecule. During replication, a random base is inserted across from an apurinic site, resulting in a 75% chance of a mutation. Deamination of cytosine and 5-methylcytosine have much different consequences for mutation because the deamination of 5-methylcytosine results in a T residue. This alters the original CG base pair to a TA base pair. Deamination of a C residue results in a U residue, which is promptly removed from DNA, restoring the original CG base pair. So this does not usually cause a mutation. Temporary, rare tautomeric shifts can cause changes in base structure that promote aberrant AC and GT base pairs. Reactive oxygen species produced by normal metabolic processes can damage nucleotides. Induced mutations are caused by environmental conditions. Mutagenic agents are chemicals or physical forces that can result in genetic mutations. Alterations in the ability of nucleotides to base pair correctly in the double helix can also cause mutations. Base analogues that are incorporated into DNA at replication can shift from keto to enol forms and alternately base pair with different nucleotides in subsequent rounds of replication. Intercalating agents such as ethidium bromide or benzopyrines distort the double helix and trigger DNA repair mechanisms which may remove nucleotides and replace them incorrectly due to the distortions in the base pairing. Ionizing radiation causes severe damage to DNA, such as single and double-stranded breaks and separating bases from nucleotides. Non-ionizing radiation, such as ultraviolet light, causes thymine dimers, which prevent DNA replication. Next up are going to be a discussion of ways that we can repair all this damage that we are constantly incurring to our DNA.